once again, CBSE class 12 student, biology. The same topic, the sexual reproduction in plant. And the topic today we have to discuss is microsporogenesis. Microsporogenesis. Simply, we call it the formation of the microspores or spores or pollen. The male sexual gamete is called the microsporogenesis. Now, what happens? We see the TS of the anther. It has somehow this type of the diagram. Once again, I made it again for you. The four lobes are clearly visible. First layer is called, I made it again. The first layer we know that the outermost layer is made up of the four layers, and the outermost layer is called epidermis. Okay, outermost layer is called epidermis. After epidermis, the next layer, this is the epidermal cell. Okay, this is the outermost layer that we call the epidermis. After the epidermis, the second layer is there, that is called the endothecium. Few layers of the endothecium is there. After the endothecium, middle layer is there. Few layers of the middle layer is there, and it is present all over the thickness, all over the lobe. This middle layer. The three layer that is called the protective layer in the microsporangia. Okay. In the process of microsporogenesis, what happens? The role of the fourth layer is that is called the tapetum. What is the tapetum? Tapetum actually consists, it provides the nourishment to sporogenous tissue. Where is the sporogenous tissue is present? Here, the sporogenous tissues are present. This part is called the all is covered with this middle layer. Okay, and after that, this type of the tapetum layer is there in all the four lobes. Okay, this is the tapetum, and inside the tapetum, tissues are present, and the tissues are called sporogenous tissue. All the tissues which are present here. Sporogenous tissue is the tissues which are responsible for the formation of the spore. Tissues which are responsible for the formation of spore. So all the tissues are present here. Okay, we make it separately. This is the formation of the spore mother cell. This is the part of the sporogenous tissue. This is the part of the sporogenous tissue. So all the tissues which are present here, we call it the sporogenous tissue. These sporogenous tissues undergo meiotic division. Meiotic division for the formation of the gamete, the formation of the number of cells, but that is the formation of the number of haploid cells to form the microspore tetrad. Now, okay, see this diagram first. This is one cell that we call the sporogenous cell having the tissue. What happened in this cell? The meiosis division takes place. Meiosis. Meiosis is the reductive division. Meiosis first occur here. This is the meiosis first occur here. And what happened due to the meiosis first? It is divided into two cell structure. It is divided into two cell structure. Two cells are formed here, and this two cell structure is called microsporidiad. This is the microspore. And what is this? This is the microsporidiad. Diad with two. Microsporidiad. Diad with two. Afterwards, the meiosis 2 occurs here. Meiosis 2. And what happened? As a result of the meiosis 2, it is divided into 4 cells. It is divided into 4 cells. 4 cells are formed here. Clearly visible 4 cells are there. Like this. 1, 2, 3, and 4. It having their own nucleus, and this is called the microspore tetrad. Microspore tetrad. So what happened in the sporogenous tissue? The sporogenous tissue which is present here undergoes meiosis division or meiotic division, and in both the step of the meiotic division, after meiotic 1, the microsporidiad is formed, and after meiosis 2, the microspore tetrad is formed. So this tetrad is formed here, and that tetrad gets separated and further forms the pollen grain. Every cell of the sporogenous tissue is a potential pollen mother cell. Okay, it is a pollen mother cell, potential to make the pollen mother cell. In the process of fertilization or in the process of the formation of microspore, the pollen mother cell, the potential to pollen mother cell to going through the process of the meiosis to form this tetrad, and this process of the formation of tetrad is called the microsporogenesis. Okay, I already told you what is microsporogenesis. It is the process of the formation of the microspore. So what happens? This is the TS of the anther. In TS of the anther, the lobe is made up of the 4 layers. Outermost layer is called the epidermis, first layer. Few layer of the epidermis is there, then endothecium is there. Then and after endothecium, middle layer is there. All three combine together to form the outer layer, which is only the protective layer, which protects the sporogenous tissue. The sporogenous tissue which is present here. What is the role of the sporogenous tissue? The sporogenous tissue is actually the potential pollen mother cell. They are responsible to the formation of microspore. And how this process is going on? Each cells which are present here separated as a microspore. Each microspore goes for the meiosis 1. As a result of meiosis 1, 2 microspores are formed. This form in the 2 part, that is called the microsporidiad. Diad is the 2. Then meiosis 2 is occurred, and as a result of meiosis 2, microspore tetrad is formed. And this formation of microspore tetrad is called the microsporogenesis. Okay, so what is the microsporogenesis? How can we are able to define it? I repeat once again, point wise. How this process is going on. I rub this all labeling because you know it very well. Okay. Now what happened? The first step, what we have to write for the note. The first step, the cell of the sporogenous tissue undergo meiotic division. To form the microspore tetrad. Cells of the sporogenous tissues undergo meiotic division. Due to meiotic division, tetrad is formed. Tetrad or microspore tetrad is formed. Due to this meiotic division, microspore tetrad is formed. Simple. This is the one cell of the sporogenous tissue. As we know that each cell of the sporogenous tissue is potential microspore mother cell. This is the sporogenous tissue or microspore. Microspore. Meiosis 1 is going on. Meiosis 1, and as a result of the meiosis 1, this single cell is divided into 2, and this is called the microspore. Microspore diet. The meiosis is completed in the meiosis second, and due to meiosis second, this cell is divided into 4 parts: 1, 2, 3, and 4, each having own nucleus, and this is called the microspore tetrad. Tetra is for the 4, so tetrad is the 4 cells are formed here. Okay, we know that every cell of sporogenous tissue is potential microspore mother cell. Potential microspore mother cell or pollen mother cell. Microspore mother cell or pollen mother cell. Pollen mother cell, PMC. So each sporogenous tissue has this ability of the microspore pollen mother cell. Okay. So what happened in this process? The process of formation of what is formed here? The substance which is formed, the thing that is going on is the formation of microspore from pollen mother cell. Formation of microspore from pollen mother cell, PMC, pollen mother cell, through the process called meiosis. Through meiosis is called microsporogenesis. Okay, what is the simple definition for microsporogenesis? Simply we call it the formation of the spore or the pollen is called the microsporogenesis. Now what is the peculiar thing is going on? Each microspore mother cell undergo meiosis to form haploid cells. These tetrads are haploid in nature. These tetrads are haploid in nature. Okay, tetrads are haploid in nature. Tetrads are haploid cells. Haploid is half number of the chromosome. Half number, just half of the parent cells. So these are the haploid in nature. They are from microspore tetrads are haploid cells, haploid in nature. Because we know that half percentage of the chromosome comes from the male organ and half percentage comes from the female organ. So it is haploid in nature. As the anther matures, when this anther going on the process of mature, this anther cells get dehydrated, and due to dehydrated, here it is built up. What happened here? Let's see here in the diagram. This is the microspore mother cell, PMC. Here it is PMC, pollen mother cell. This is microspore mother cell. What happened here? It's going on for the meiosis one, and because of this, the dyad is formed. Dyad is formed. Then here, this is the one cell. They are taking dyad cell is there. Here meiosis is going on, and meiosis is going on. Dyad is formed. Here it is converted into tetrad. Here the tetrad is formed. When this tetrad gets mature, what happens when this tetrad gets mature? Each tetrad is separate to form. Each tetrad gets separate to form the pollen. जो भी tetrad है, this is the potential microspore mother cell. PMC or MMC microspore mother cell gets separated like this, and in each cell the meiosis 1 is going on, due to which the dyad is formed. This is the microspore, dyad, microspore, dyad. Then meiosis completed in the step called meiosis 2. Because of that, the tetrads are formed. This is called microspore tetrad. Okay. Now what happens? This microspore tetrad is further converted into pollen. How the pollen is formed? This is the part of the next topic, that is the pollen grain, its formation, structure and development. Okay, what happens when the pollen is formed? This answer gets dehydrated, and because of the dehydrated, the pollen, this part of this the pollen answer, this part of the answer set gets burst, or that dehiscence is occur there, and because of that, all the pollens come out from this part. All the pollen which is formed come out from this part. In one anther sac, large number of pollens are present. Large number of pollens are present which are released after the process of dehiscence. Okay, so first move towards the formation of pollen. The next topic is the formation of pollen. How the pollens are formed and what is the structure of pollen. Pollen is the male sexual unit. Pollen is the result of the male sexual unit, or it is the male reproductive unit, or the male gamete of the plant cell. Okay, so let's move towards the pollen grain. Pollen grain. Pollen grain. Pollen grain, these are the microspores. Actually, microspore tetrads develop to form the pollen grain. Okay, so what happens? It is developed or mature. It is generally spherical in shape. Generally spherical or oval. Spherical in shape. Okay, it is spherical. It is actually microspore. Actually, microspore. Microspore. Okay, it is actually microspore. It's generally spherical. And what about the size? It is 25 to 50 micrometer in diameter. It is 25 to 50 micrometer. Micrometer in diameter. In diameter. Just think about the size of the pollen grain. It is 25 to 50 micrometer in diameter. It is haploid cell. It is haploid cell. Okay, why it is haploid? Because it's a one sexual unit. It's only male sexual unit. The prominent two-walled structure is there. What about the structure of the pollen? Let's see here. The structure of pollen. It is made up of prominent two-wall. Okay, it is made up of prominent two layers. And what the layers are called? What this layers are called? The outer layer is called exine. Outer layer is called exine, and inner layer is called intine. Okay, two layers are there. The outer layer is called the exine. Inner layer is called the intine. In exine, the outer layer, in some spores, some spine-like structure is present. It is not compulsory for each and every spore. Some spores contain this spine-like structure or exine. This spine is not very much compulsory for each and every pollen. If spine is present, it is called rough pollen grain. And if spine is absent, then it is called smooth pollen grain. Okay, it is not necessary that each and every pollen grain contains this spine, but somehow spines are present on the exine. Inside this pollen, large number of cytoplasm is present. Cytoplasm, which is content of each and every cell. The cytoplasm is present inside this. Inside this, large number of cytoplasm is present, and one shapeless nucleus is also present here. Shapeless nucleus is also present here. So what about the structure of the pollen? It is composed of the two layers. One is called the exine and one is called the entine. Exine is the outermost layer and entine is the innermost layer. In exine, some grooves are present. This is called germ pore. Okay, some grooves are present. That is called the germ pore. In exine, some spines are also present, but that is not the compulsory structure of the pollen. If the spines are present, then it is called the rough pollen grain, and if the spine is absent, then it is called the smooth pollen grain. Now, more about this pollen grain. So, what happened in the pollen grain? We have to talk about the exine and entine. What is the role of this exine and the entine? Exine. First is the exine. That is the outermost part, outermost layer. It is very much important. Why it is important? What is the importance of exine? Exine is made up of one material that is called sporopollenin. Made up of sporopollenin. This sporopollenin, it is very much important substance. It is organic matter. It is organic matter which form the outermost layer of the pollen, that is the part of the exine by which the exine is formed. This layer, the sporopollenin, it withstands with high temperature. Okay, it is able to survive in high temperature. Able to survive in high temperature. High temperature. Not only in high temperature, it is able to survive in high temperature. Strong acid, and strong alkali also. Okay, means no substance is able to destroy it. Even no enzyme acts on it. No enzyme acts on it. No enzyme acts on it. इसके ऊपर किसी एंजाइम का असर भी नहीं होता. So it is preserved as fossil. It is preserved as fossil. It is very much hard. कि बहुत हार्ड है, हार्डेस्ट नोन सब्सटेंस है. Sporopollenin hardest substance भी है, and it is made up of the organic material, and it is not single substance is there. No temperature, strong acid, strong alkali, enzyme, nothing is acted on it. कोई भी इसके ऊपर effect नहीं डालता. It means it is the most resistant substance. Most resistant substance. Out of all the resistant substance on the earth, the sporopollenin is the most resistant organic substance. Okay, so sporopollenin is important. Why it is important? Because it is the most resistant organic substance. This is about the exine. Now, what about the entine? What about the intine, the innermost layer? Okay, now intine, the second layer, out of the two layers, the inner one is called the intine. And what is the importance of this intine? This is the thin continuous layer. This thin continuous layer. Thin continuous layer. Exine is not the continuous, some furrow or the pores are present in exine, but in intine it is the continuous layer which is made up of the cellulose and pectin. It is made up of cellulose and pectin. Okay, it is made up of cellulose and pectin, and it is continuous also. It is continuous layer also. Exine is not a continuous layer. Okay, what happens about the third layer? This is the exine. This is the intine. The third is the germ pore. Germ pore. Germ pore is the pore present in exine. Some pores are present in the exine that we label as the germ pore. So germ pores are the pores which is present in the exine. And what is the importance of germ pore? As we know that exine is the hardest substance. It is the highly resistant substance. It is the substance which is present inside the exine is unable to break that hardest layer. Then how the process is going on? How the pollen tube is formed for the process of the pollination? The pollen tube is formed from the germ pore. What is the importance of the germ pore? Germ pore give rise the pollen tube. It gives rise the pollen tube. The pollen tube. Okay, this is the importance of the germ pore. Because all the exine is the hardest substance, it is highly resistant to dissolve it or destroy it. So the germ pore is there. Germ pore, this is the germ pore. Okay, it develops like this. This is called the germ groove or furrow. Groove or furrow. Groove or furrow. And this further divide into the tube. That is called the pollen tube. Pollen tube. Okay, that is about the germ pore. That is the importance of the germ pore. Germ pore in monocotyledon and dicotyledon. So different type of the germ pores are there. In monocotyledon, the uniporate germ pore is there. Uniporate. Uni this one in monocot. This germ pore is uniporate. Uniporate. And in dicot, it is triporate. Tri is for three. Triporate. Triporate. That is importance about the germ cell. This is the importance about the germ pore. Okay. Now, what happened inside the pollen? Inside the pollen, one plasma layer is there, which further grow of the pollen tube. Cytoplasm is there, and nucleus is also there. What happened again in this pollen tube? In this pollen, what happened? It is going for the mitotic division, and due to the mitotic division, two parts are formed. Okay, this is the pollen. Okay, mitotic division goes on in the pollen, and this due to this meiotic division, not the mitosis is the meiotic division. It is divided into two, but that is not the equal part. It is divided into two unequal parts. The largest part is called the vegetative part. Vegetative part, and the smallest part is called the reproductive part, or it is called the generative part. Generative part or generative cell. This is vegetative cell. Vegetative cell, which is responsible for the formation of the pollen tube, etc., etc. Generative cell is responsible for the formation of the spores or formation of the main gamete. This stage is called two-cell stage. This stage is particularly called the two-cell stage. Okay, this is the two-cell stage. What happened in the two-cell stage? The two parts is present. Vegetative cell and the reproductive cell. That two parts is present. Pollen grain separate from this anther, comes out from the anther after maturity. But in 60% of the angiosperm, the pollen grains come out in this stage only. Okay, in 60% of the angiosperm, all the pollen grains which are comes out after the maturity of anther. In the maturity, the anther is dehiscence or the breakdown is going on the anther, and all the pollen grains separate out. In 60% of the angiosperm, the pollen grain is separated in 3 cell stages. Okay, the pollen grain is separated in 3 cell stages. This is the in 60% angiosperm. Okay, we write it here. If we have to rub this. Okay. Now what happens in this pollen? Pollen goes in the process called the division. What division process is there? That is called the mitotic division. What happened due to the mitotic division? Okay, mitotic division is going on. Mitotic division or meiosis division. Okay, due to this mitotic division occurs in pollen. Okay, what happened due to the mitotic division? It is divided into 2 parts. 2 cells are formed. Okay, 2 parts or 2 cells. 2 cells. What is the name of this 2 parts? The largest part is called vegetative part. Vegetative part or vegetative cell, and the smaller is called generative cell. Generative cell. It is small in area. Okay, generative cells. 2 types of the cells are formed: vegetative part cells and the generative cell. What about the vegetative cell? What is the structure or condition of the vegetative cell? It is bigger. This vegetative cell is bigger as compared to the generative cell. Vegetative cell is bigger. It contains large amount of food reserve. Okay, it contains large amount of food reserve, food storage. Why the food is stored here? It provides the food for the developing gametes. It provides the food for developing gametes. So it contains the food, storage of the food. Food reserve is there. Okay. Also, it has the nucleus, but this is irregular shaped nucleus. Irregular shaped nucleus. This is the peculiarity of the vegetative cell. Now, what about the generative cell? Generative cell is small as compared to the vegetative cell. It is small as compared to the vegetative cell. It is small in size and it is floats in cytoplasm. The fixed position is not there. It is floats in cytoplasm. Floats in cytoplasm. Okay, it floats in cytoplasm. Cytoplasm, whose cytoplasm? Of vegetative cell. Cytoplasm of the vegetative cell. So that is the peculiarity of the vegetative cell and the generative cell. Generative cell further divide to form the male gamete, and the vegetative cell further going to form the pollen tube, and the generative cell further going to form the male gamete. Okay, when it is divided into 2 parts, this is the 2 cell stage, and in 60% of the angiosperm, pollen gets separate in this stage. Okay, dehiscence is occur. Okay, in 60% of the angiosperm, but rest of the 40% angiosperm goes for 3 cell stage. I write it again. Okay, what happened? This is a 2 cell stage which occurs in 60% of angiosperm. Angiosperm is the flowering plant. 60% of the angiosperm release the pollen grain in this state, the two-cell state. Releases the pollen in two-cell stage. Okay, that is called the process of dehiscence. But what happened of the rest 40%? In rest 40% of the angiosperm, this pollen goes for the three-cell structure. What happened in the next 40% of the angiosperm? 60% is two-cell. In 40% of angiosperm, this cell. Okay, the first division is going on, and here two-cell structure is formed like this. This is the vegetative cell. This is the generative cell. The generative cell further divided to form two, further divided, and this divided is the formation of two male gametes. This is the male gamete. And this is called the three-cell structure, three-cell stage. Okay, this is the three-cell stage. So 40% of the angiosperm release the pollen in three-cell stage, and 60% of the angiosperm release the pollen in two-cell stage. So this is about the pollen grain. Now, one more important thing about the pollen grain. The branch in which we have studied about the pollen grain is called palynology. Palynology. What is palynology? It is the branch in which the pollens are studied. Study of pollen grain. The study of pollen grain is called palynology. Okay, this is very much important. You have to remind it also. One more thing is that pollen we know very well. You go and plug one flower, touch the central part of the flower, you get something in your hand, some yellow substance in your hand. That is actually the pollen. If you have the microscope, release your hand in the water, keep that water in the slide and see in the microscope. It saw the shape and size presence of the pollen grain there. The pollen grain is very much present in all over the environment. But every time the pollen grain is not important for us, not beneficial for us. Anyhow, pollen is important. Why pollen is important? It is used as a nutrient. It is energy source. The person who are doing the hard labor, such as players, etc., they use the pollen. They use the pollen, that is the users of pollen. Okay, users of pollen. Pollen is used by hard worker persons. They use the pollen in the form of the tablet or in the form of the syrup. They use the pollen as it is provide energy. Highly nutritive. It provides energy. Okay, provides energy. So it is used as a food also. Pollen is used as a food also. But you know, few people are there who are allergic towards the pollen. Allergic, they show some symptom of the allergy such as sneezing, coughing, and something like that, because when they come in the contact of the pollen. So pollen is very much allergic also. One important flower is there, one plant is there that we call the parthenium. Parthenium is very much present in all our locality. You know it very well. You saw it also. It is harmful for us because it is highly allergic. It provides some lung disorder like bronchitis, coughing, okay, irritation, etc., etc. And that is called the parthenium. Parthenium is a plant. It is actually the weed. It is grown with wheat also. Wheat, the main crop of India, it's grown with the wheat also. In common, we call it the carrot grass. Gajar grass. Okay, the pollen of this carrot grass is highly allergic. Okay, it releases some bronchitis effect, bronchitis or the lung disorders from that. Sometimes it generates the fever also, and that is called the hay fever. What is hay fever? The fever generate due to effect of pollen. Okay, this is about the pollen. Okay, what happened? Must remember that diagram, the first diagram that we have formed. The first diagram, what happened? We saw in the TS of the anther, the four lobes are clearly visible. I made it the cane. This is the TS of anther. Okay, what happened here? Here is the microspore mother cell, that is a potential microspore mother cell is there. This microspore mother cell gets separated here to form diet, microspore diet. This microspore diet gets converted here into microspore tetrad. Okay, this microspore tetrad further developed to form. This microspore tetrad further developed to form pollen. Okay, further to form pollen, and at the maturity, this anther gets dry, dehydrated, and because of the dehydration of that, the dehiscence occurs from this part. Dehiscence is the breaking of the anther sac. The dehiscence occur in this part, and all this pollen gets separated out from here. They are responsible for the process called the pollination. All the pollens come out from here, and the pollens are responsible for the process of pollination. So this is all about the microsporogenesis and the formation of pollen grain. So in male reproductive part, what is going on in the class? Firstly, we saw the structure of the stamen. Then we saw the microspore, then the process of formation of microspore, that is called the microsporogenesis. Afterwards, the pollen grain is structure and the shape and somehow little bit the function of pollen grain also. Afterwards, we have to study about the pollination and all in my next